All right, in this unit, we're going to be focusing on DNA and RNA, looking at structure, looking at how they are synthesized, focusing also on scientists involved in discovery and in transmission of DNA from species to or from cell to cell, those types of concepts. So let's first get started by looking at the science behind it, those scientists, the experiments they did, and how they made these discoveries. First, let's look at Frederick Griffith. Back in the late 20s, he carried out an experiment where he took a live version of a smooth species of bacteria and crossed it with a species of mouse, and it killed the mouse. So he took a different type of bacteria, a rough species of bacteria, crossed it with the same species of mouse, and the mouse was able to live. So he thought, okay, well, let's try dead versions of this bacteria. So he took a dead version of this smooth bacteria. The mouse was fine, it was able to live. So he tried one more thing. He said, what if I did a live version of the rough bacteria, added it to or combined it with the dead version of the smooth bacteria, since those were both versions that the mouse was able to live, and then cross it with the mouse DNA, what would happen? Well, the mouse still died. What was the conclusion for this? Um, when he looked at the reason behind why the live version of the rough bacteria and the dead version of the smooth bacteria being combined, why it killed the mouse, he found that there was a part of the live rough bacteria that was able to make the dead smooth bacteria that he transplanted with the mouse. He was able to see that it caused that dead S bacteria to become dangerous. This process was later deemed or called transformation, which what happens in transformation is a change is caused when a cell takes up a foreign DNA. So what happened was that dead S bacteria was able to take on some of the DNA from the live R bacteria, or excuse me, the live R bacteria was able to take on some of the DNA from the dead S bacteria, making it dangerous, which killed the mouse. So this happened in the late 20s, this transformation process where DNA could take on characteristics of other DNA. Around the same time, early research, Erwin Korgoff came along. And he did studies on the makeup of DNA structures back in the 50s, and he found that there were equal parts of amino acids that made up certain strands of DNA. He found that adenine and thymine, there were always equal amounts of that, and he also found that cytosine and guanine, there were always equal amounts of those. Not actually knowing how they bonded together, though, but he did find that by looking at some early experimentation, he was able to find that these amino acids, adenine and thymine, usually had equal amounts to them in DNA, as well as cytosine and guanine. And then shortly after Kargoff's experiments, Rosalind Franklin, probably one of the... Okay, so what Rosalind Franklin, around the early 50s, she used X-ray technology. Okay, she took pictures, these crystallography pictures using X-rays, using gamma rays for the X-ray, so using radiation to take these X-ray pictures to determine what the structure of the DNA was. Okay, this was Rosalind Franklin and Maurice Wilkins. Now, she did this in the early 50s. Back in the early 50s, women were not there wasn't equal rights. Let's look at it that way. So the discoveries she made were taken by Watson and Crick about a year later. They took what Franklin found. They kind of credited it as their own information. They didn't really give her any type of credit for it. And they used those pictures to help determine, they took it a step farther, and they helped determine the actual structure of DNA in 1953. So the crediting for the structure of DNA, that double helix, was given to Watson and Crick, but they were the ones that took the original experiments that Franklin did to as their own, in essence, and was credited for finding that structure, that double helix structure. Okay, they did find that there was this double helix, this two-sided spiraling structure of DNA. Won a Nobel Prize for their discovery, one of the biggest discoveries in DNA history. So let's look at, start getting into this DNA, looking at the structure based on what they found back in the early 50s. Remember our organelles. There's one big organelle that classifies whether or not it's a prokaryote or a eukaryote, and that's the DNA, or that's the nucleus. And the nucleus is where your DNA is going to be located. Our prokaryotes have DNA as well, or they have genetic material. However, it's just housed within the cytoplasm of that cell. There's no extra. It's like a nucleoid region. It's a region of an area within that cell for the prokaryote, but there's no nucleus that houses that structure. So only in the eukaryotes will there be that nucleus that houses the DNA. Okay, within the DNA is miles and miles long if it were to be stretched out, so it has to be compressed into structures called chromosomes. Those chromosomes are going to hold the DNA structure, then we know what that DNA, or we're going to look at what that DNA structure is actually made of. And we should remember when we looked at macromolecules that this particular structure here is known as a nucleotide. And it is made by nucleic acids. Okay, so that building block of nucleic acids then would be that nucleotide. Okay, we know what our DNA is. We know that it is the material that stores the genetic information for that particular organism. Okay, it's like a really large book, and it has within that book all of the instructions for all of the different codes and processes for that organism, whether it be codes for building proteins, and those proteins help then code for physical characteristics, for genetic makeup, for internal makeup. All of those different characteristics that make up an organism are coded within that book of genetic information. We'll get to this later, but a whole book of genetic information on one organism is known as the genome of that organism. We've looked at proteins before. We know proteins are used for a lot of different things. We know that they are the code for making all those different proteins are found within the DNA structure. We know that enzymes are also important for these proteins. Everything from transport across the cell membrane, and everything from energy makeup for the amount of energy through cell respiration, for building muscle mass, for a lot of different types of processes for an organism that proteins are important for. All right, so this DNA structure, we know DNA is the large polymer, okay, that's made by those nucleic acids. So a nucleic acid, its monomer would be a nucleotide, its polymer would be the DNA or the RNA. Okay, so this large structure of DNA is made up of monomers, which are nucleotides. We've looked at nucleotides before, but we're going to look at them again. We know that there are three basic parts to a nucleotide: its sugar component, its phosphate group, and then its nitrogen base. And each one of those things has a different job for the structure of DNA. The sugar part of DNA is called deoxyribose. Right, it is a 5-carbon sugar. It, along with the phosphate group, and a phosphate, chemical formula for a phosphate is PO4, 1 phosphorus, 4 oxygens makes a phosphate. The sugar, deoxyribose, and that phosphate group combine together to make the backbone to help build the structure of the DNA strand. And then the nitrogen base, it's a carbon-based structure as well. The nitrogen base is going to be where the bonds occur to build the connecting points to make the double helix. It does have nitrogen in it. It also has carbon. But the type of nitrogen makeup will determine the type of nitrogen base. And like I said, that's where that's what helps bond together these two separate pieces to make that double strand. So here's the basic structure of a nucleotide, a picture structure or a chemical formula structure. We've seen this before. We see our phosphate, our phosphorus with our 4 oxygens. We see our 5 carbon sugar. Okay, and then over here we'll have a type of nitrogen base. It'll have nitrogen in it, it'll have carbon structure in it, and depending on the arrangement of this will determine what nitrogen base it is. So there's your phosphate group, there's your sugar. For DNA it's deoxyribose. When we look at RNA, the sugar would be ribose. This is your 5 carbon sugar. And then your nitrogen base. Okay, then this whole structure will make your nucleotide. Alright, now let's look at the 4 nitrogen bases that could be used to form a particular nucleotide. Remember, these were discovered by Chargaff, as far as not discovering them, but discovering their relationships. And we know that there is adenine, and that there's guanine, cytosine, and thymine. Those are the 4 nitrogen bases, and they are grouped into 2 categories. And we'll look at what these categories mean later, but your adenine and guanine are going to be considered purines. And then your cytosine and thymine are going to be considered pyrimidines. Now they bond together. Remember, this is Chargaff's discovery. They bond together. We know that cytosine and thymine usually make bonds together because they're the same type of nuclear, or they're the same components of nucleic acids or nitrogen bases, because they are the pyrimidines. And then adenine and guanine will also go together because they are both going to be considered purines. Similar makeup. They go together. There is equal amounts of each one within a particular DNA strand, and then their bonding together is what forms that double helix. Okay, so if you look at a picture of a double strand, the backbone, this outer structure, is going to be made up of your phosphates and your 5 carbon sugar. So the phosphate, 5 carbon sugar, phosphate, 5 carbon sugar, and they will be bonded together. The other side as well. And then the middle part, where the actual structures connect to each other, is where your nitrogen bases meet, whether it be an adenine with a thymine, or a guanine with a cytosine, and so forth. And as I mentioned before, the rules, these base pairing rules, we know that cytosine will always bond with thymine. It'll use a triple bond to make that happen. Okay, and then adenine will always bond with thymine, and there will be a double bond. Your purines use double bonds, your pyrimidines use triple bonds. The type of bond that holds these together are called hydrogen bonds. Hydrogen bond is a very stable, very strong type of bond. It also can be easily broken, but is a very good type of bond, especially for containing DNA structure. Now, if you were to look at this picture, we know that the sugar will be the 5 carbon, 5-sided sugar, your phosphate group. These two make up the backbone. Then we know guanine would go with cytosine, cytosine with guanine, on down the list, and those would be your nitrogen bases. If you were able to write down all the bases for that came out of just one cell, okay, it would take up to about 1,000 stacks of phone books to make up just one set of bases from a cell. If you were to unravel all your DNA from all your cell, it could stretch to the moon and back 6,000 times.